Hey everyone, I'm Elad from Astrolabe Diagnostics, and welcome to Single Cellmates. In this web series, I interview influential and interesting figures in the world of immune monitoring. My goal is to learn about their background in the field and hear about their thoughts on current and future challenges. Our guest today is Dr. Susan Raju Paul, a research fellow at the Vaccine and Immunotherapy Center in Massachusetts General Hospital. Susan comes from clinical medicine and has transitioned to research in 2014. She investigates how understanding the immune profile allows us to predict disease outcome and facilitate therapeutic advancements. Hey, Susan, thank you for joining us today.
Thank you for having me, Alad.
And you're a clinician by training. You then transitioned to the bench, and eventually you integrated bioinformatics as well. Could you tell us about your journey and what prompted your move at every step of the way?
Yes, so I wish I could say that this move was entirely calculated, but that isn't the case. I wanted to entirely be in clinics. I love clinics and love patient care. I love the element of being able to serve out my clinical interest in taking care of patients and anybody across the field. So I enjoyed very much being in the hospital setting, and in India especially, which is where I got my training. I enjoyed being with a lot of infectious disease cases, and we have quite a few infectious disease cases in India. So being able to take care of them was something I did enjoy. To advance my training, I initially came to the US in the hopes of getting into residency, but I also wanted to get into a good residency program. So I thought of doing some research before that, as I didn't have a good background in research. But turns out, as I got myself a little into research, I quite liked it. I enjoyed the intellectual stimulation, the challenging questions, and the fact that we actually have to seek out some answers that we don't have already. And much like diagnostic medicine, but in a different way. And I quite liked it. So that's why I decided very recently, after being in research for about 5-6 years now, very recently, that I would like to continue on a path of exclusively research and not in clinical medicine for now, at least. So that's how I shifted from clinics into research. But however, my research definitely has, I always think about the clinical background because I've treated the patients, I've had the opportunity to see them. So I always think of, okay, if we are thinking of a drug, if you're thinking of a new way of administering a vaccine, how feasible is it? Like if you think of doing something, giving a vaccine to a child, you should remember it's a child and how feasible it is. So I always think of the clinical part about it. So translational research is something I very much enjoy. As with regards to bioinformatics, that I got into it exclusively, or almost exclusively, because of mass cytometry, which we both share in common, our interest in it. So when I started off, it's an amazing tool, but one of the things that comes with CyTOF is it's a very high dimensional platform. Excuse me. It's a high dimensional platform. So to be able to analyze the data from CyTOF, you definitely need to have some knowledge into bioinformatics. When I started off, I did not have as much knowledge into the depth of knowledge you would need to be able to analyze some of this data. But at the same time, it's not about learning. It's not about knowing everything, but knowing enough to be able to analyze your data and knowing who to go to if you need to do further analysis. So I ventured into it because of mass cytometry, and yes, I've been around now.
Yeah, and I'm pretty excited to hear that you're sticking to research for now, because I've seen some of your work and it's quite amazing.
Thank you.
In your opinion, as a clinician turned researcher, what should be the role of bioinformatics as part of the training of clinicians, if at all?
So one of the things I tell all our students is, in the coming years, knowing coding or knowing a programming language is very, very helpful, because the truth is we have a wealth of data that's coming our way, data that's collected in clinics through patient care, data that's collected through research, and bridging those together always helps us get a more concise, a more coherent description of what can be the next option for the patient. So to be able to bridge that much of information together, you do need some level of analysis and bioinformatics. And you also need to understand statistics a lot more. We do have some level of statistics in med school. It's not a course a lot of people like to take because it's a little difficult. So yeah, I would definitely think it's very much needed in the coming years because of how much we can acquire if we were to bridge clinical knowledge, research knowledge together, which is a wealth of data.
Yeah, that makes perfect sense. And from my perspective, at least, it is, I think we will always need the bioinformaticians who are experts on it.
Yes.
With that said, it's a tool. It's like statistics, it's like a pipette. So you should be able to know how to use it just so you can communicate with the expert. And also sometimes you just want to do something small or contained, and it's really helpful to be able to do it on your own.
And it also helps you understand, even if bioinformaticians are very useful because they know the field really well, but to understand what they are talking about, you kind of need to have some baseline understanding on the field as well. So.
And thank you for acknowledging that I'm useful. I really appreciate it. We met in Boston last year, where you presented a compelling perspective for utilizing immune profiling in the context of infectious diseases. And I think that a lot of the site of work is immunotherapy or similar applications. In my opinion, some of the most exciting work is actually in this field of, again, infectious diseases, Zika, dengue fever, typhoid, and so on. In your opinion, how can this technology be used to help patients?
Yes, so that's a very interesting question and one that I always think about, and we often discuss it in the lab. So we have patients who in clinics, a lot of them who, for whatever infection it may be, it's not that each patient has the same response. Some patients, when you give a drug, they have a specific set of responses which may be good. Others have moderate responses, you take a little longer. And then some others sometimes don't respond at all. Now, there should be something that's there in the baseline that differs in each of those patients. Yes, we always talk about comorbid or underlying conditions, but what if it is that it's something in their immune system, something that if we could target to alter the response, to better the response, so that those who aren't responding to drugs as well can respond. So understanding what the baseline immune response of patients is would help us understand why these patients respond differently after any kind of infection. So I think in any kind of it's always helpful to understand how each patient or how people in general respond to different bacteria, viruses, or any of the microbes. But there's also a greater utility in therapeutics, like how do you alter or how can you understand who is going to respond better for which drug? So, and CyTOF or any multidimensional tool would help with something like that.
And I actually want to dig a bit deeper into your last statement. In your opinion, what is the future of immune profiling in the clinical context? Do you think there's going to be direct applications in the clinic, or is it going to remain the purview of research where we try to understand the infection and use that to develop better drugs?
So I think this is obviously my opinion, and I want to state that. I think there is usefulness in the clinics in understanding, because with any tool, specifically CyTOF or RNA sequencing, or you have a wealth of data that comes in. Now, profiling a wealth of data for each patient in a tertiary center would be easy, but it's not as much in a secondary or a primary care center. But if you have this wealth of information, which you can then winnow down to be able to isolate, okay, these are the specific transcripts, or these are the specific markers that you want to look for, which enable you to distinguish between good outcomes versus bad outcomes. Those specifics can be then translated into secondary and primary care centers, so you can have a larger clinical implications. Mass cytometry in large, or like any of these higher dimensional tools in large, may not be readily usable in the context of a secondary or primary care center, versus in a tertiary care center is obviously easier.
Yeah, I agree with you that I don't imagine the average PCP having a CyTOF in their clinic.
Exactly.
But I think that maybe from a secondary care perspective, it might be maybe not a CyTOF per se, but I think that specialists, I can envision a future where specialists prescribe an immune monitoring diagnostic to a patient.
Yeah,
and maybe PCP as well. Yeah, go ahead.
No,
Sorry.
So it's a good question. I think my thought is always that yes, we could have a diagnostic, but do we need a diagnostic that comprises 37 markers, Or does the 37 markers help us to come down to like 5, which can then be translated into flow cytometry that's very easily doable in a lot of these centers?
Yeah, I agree with you. I think that the pipeline that is shaping up is that you use the CyTOF for the hardcore discovery, and you can use the results from that to design a panel, a 5 to 8 color panel. And that's something that the major flow providers, Quest Diagnostics and LabCorp, they can build a panel
Yes.
around that. One of the projects you've been working on involves a vaccine for
Yes.
Coxiella burnetii. Did I pronounce it
Coxiella burnetii.
correctly?
Yes, you did.
Could you say it
Coxiella
again?
burnetii. Yeah.
Coxiella burnetii. Thank you. Could you discuss the pathogen, the symptoms, and the importance of the
Yes.
vaccine?
So Coxiella burnetii is what you would call a hardy bug in that it doesn't die very quickly. So it's a bacteria, it's an intracellular pathogen, and it has the characteristic immune responses of intracellular pathogens. When you look at infection, it's a zoonotic disease. So those who do get infected, most of them are asymptomatic. And if you do get an infection, and if you do become symptomatic, you can have like flu-like symptoms. So most people who get infected with Coxiella burnetii don't have very acute symptoms. You can usually resolve. It's self-limiting. If you take rest, it usually resolves. And in some cases, you need antibiotics. Some of those patients, though, go into chronic cases where you have diseases where the bacteria can dwell into valves and cause endocarditis. So that's one of the more common chronic conditions of Coxiella burnetii. But usually it's not a disease that causes a high mortality. However, it is something that has a high morbidity in that the chronic disease might require long-term antibiotic treatment of up to about 2 years. So that's not easy. That's a lot of economic costs that we're looking at. Now, in our case, it was a project that was contracted by the Defense Threat Reduction Agency, by the DETRA. The reason being that US troops, when they go to certain specific regions in the Middle East, they found that some of them, and these are published articles, that a good percentage of those troops that come back seroconvert and become positive or have been exposed to Coxiella burnetii. So in which case, because of which the defense is obviously interested in getting a vaccine against the disease so that you can vaccinate the troops before they're sent out into these endemic regions. Now, right now, there is a vaccine available, but it has a component where if you have had any prior exposure to Coxiella burnetii, then you could be reactogenic, because of which the FDA hasn't approved it yet. So a lot of us know sometimes, like, because it's like flu-like symptoms, sometimes you don't realize you've had the bug because you might have just had a little bit of body ache, so you don't know if you've been infected or not. So anybody who can get the vaccine that's currently not FDA approved and hence not available in America, it's only in certain other countries in the world where it is approved. If you were to get vaccines in those countries, you need to have a skin prick test to look for reactogenicity in any way. And if you don't have reactogenicity, then, or look for any exposure. So if you don't have any exposure, then only can you get the vaccine. Because if you have had any exposure, then there is a chance that when you're given the vaccine, you could be reactogenic. So, which is why we looked at getting a new vaccine that would not have the reactogenic component, in that it could be given to anybody and everybody without the need for testing for prior exposure through any skin prick or blood test. So that was why we looked at the disease and we looked at developing a vaccine. So we were contracted to look at getting a vaccine against Coxiella burnetii. Now, my role, I forgot the second half of your question, so could you repeat that?
Actually, that's the question.
Okay.
And I'm curious, is that, just taking a step back, I think epidemiology has become a centerpiece of the news lately. This process you described, where American troops go abroad and come back with an unusual pathogen, is that a common occurrence with other diseases as well?
I think a lot of them, you have vaccines. Like when any of us travel abroad, we have, like, travel medicine is a topic of interest because when you go to certain places, you do have vaccines that you need. Specific to the troops, I wouldn't be able to comment, but we do have travel medicine for the same reason, that when you go to certain regions where you have diseases that are endemic, you do need to take either a drug to prevent the disease or a vaccine to not contract the disease. So,
Got
yeah.
it. And yes, actually, now that we know more about the pathogen, can you tell me a bit about your specific work in the field? What experiments are you running? Which technologies are you employing?
So yeah, so if you wanna make a new vaccine, it sounds very simple. Can do it in a few days? No, definitely not. So the vaccine, when it comes to developing a new vaccine, one is how do you test that the vaccine works? So we employed two different. We looked at it in a two-prong approach. The first was if a vaccine works, what would be the signature? So to identify those signatures, what we looked at is what's a natural infection, as in or a natural vaccine. So that would be anybody that got an infection. And so we identified a cohort of people in the Netherlands who did have infections with Coxiella burnetii. And so what we did was we profiled them, and they would be what you would call a natural vaccine, to understand what are the signatures in the body in response to a natural vaccine. Because
And
you
when,
would want
and when you say profile them, what do you mean by that?
we profiled them using mass cytometry. So we looked at immune profiles using mass cytometry. And the hope is that the new vaccine that you're creating would have a signature which mimics what is a natural vaccine, which is an infection. So to understand what would be protective. Did that make sense?
I think so. Just to read it back to you, you have a cohort of former patients,
Yes.
and you can profile them using the CyTOF, identify the different cell populations in that cohort, and you compare that to another population that you have vaccinated with this tentative
Yes,
vaccine.
but we haven't yet vaccinated because obviously a vaccine hasn't yet been approved for something like that. But the hope is that when you do vaccinate, you do have a signature
Okay.
in those patients or in those donors who do in those volunteers who do get the vaccination, that post-vaccination they have a signature that's similar to those who have a natural vaccination or an infection. So to understand what would be the signature, we did look at immune profiles in those who did develop infection to Coxiella burnetii. So that was the human part. The study, we've completed it, the analysis is underway, and we should have a publication out late fall. The other was with regards to any vaccine, we all know we need to test it in animal models. So to test it in animal models, the first thing that we did is there is a current vaccine that is useful for that can be used against Coxiella burnetii in animals. So we vaccinated animals with those with the vaccine and looked for post-vaccination signatures. So whatever those post-vaccination signatures were, we tried to see if we can mimic those post-vaccination signatures, which then correlate a successful vaccination with a new vaccine. So we had both a human study and an animal study, and the animal study was recently published. The print is not yet out, but we should have it released soon.
And if I understand you correctly, the structure of both studies is very similar. In both cases, you have an immune population, which could either, in the case of humans, have been exposed to the pathogen, in the case of mice, has been vaccinated. And then you have a second population, which you would like to compare to that first population in order to identify, to see if you can identify the same
Yes,
signature. Did I get it
the
right?
essence of what you're trying to do is identify how do you know, or you're trying to answer the question of how do you know that your vaccine worked? Outside of challenging somebody with Coxiella burnetii, how do you know, is there a way to know in their blood system that the vaccine that you've given has worked? So we tried to answer that question using mass cytometry by profiling in the case of humans with those who've had infection in the past, or in the case of mice or in animal models post vaccination with the current available, what's the signature in them.
And hypothetically, is this one of the signatures that could be reduced into a flow cytometry panel? So if you vaccinate someone in the future, could you test and see if it works?
Yes, so that's what we are attempting to do. So one of the things is to narrow it down to 5 or 6 markers or whatever can be accommodated. So we know up to about 10 markers can be accommodated easily in a flow panel. So up to 10 markers in the, yes, that's what we're trying to do, reduce the cytometry panel into that.
And looking at the big picture, once that study is completed, and let's say you're happy with the results, you have a signature that is promising, what would be the next step in the vaccine development? Where would this research feed into?
So obviously the next step would depend on whether the vaccine works or not. Right now, I'm not at liberty to speak about that. But if we're talking about the vaccine does work and it goes into clinical trials, then it could be that in clinical trials, you can draw blood from these patients and profile them through cytometry or flow cytometry, ideally, because you would hope that you can shrink it down to 10 markers versus 37. So profile all of those patients who do get your vaccination and see if they have a signature. So which would help you understand, without giving a challenge, is this vaccine protective?
Yeah, that makes sense. And according to your description, this sounds like a pretty complex mass cytometry experiment. And I imagine that you have dozens or maybe even more samples from either patients or mice. In your opinion, what is the most challenging aspect of a study of this magnitude?
So I think with any high dimension study, the biggest challenge is how little we understand, or how little I understood initially, the need to optimize really well. If you set yourself up well, your downstream analysis can go really well. If you don't, your downstream analysis can take a really long time. So the biggest thing is making sure you optimize all your parameters, optimize the conditions in which you collect the cells, optimize the conditions in which you stimulate them, optimize your antibodies, tighten them, make sure you have the same lot that you use throughout the study, and as much as possible to reduce batch variation, do all the labeling at one go. So optimizing will make your downstream analysis much easier, because otherwise you'll have to think about things like batch effects and normalization.
And could you maybe elaborate? Let's say a researcher is facing her first high complexity study. Could you share some tips or some pitfalls to watch out for in this process of optimizing the experiment?
Yes, definitely. So when you start off with mass cytometry experiments, the first is start off on a smaller scale. Make sure that instead of starting off with 100 patients, start off with, I'd say, about 20, and see if you can make it work. So for that, the first things you need to know is, okay, what are your cells? Are they all coming in the same condition? So you take blood out of, if you're talking about a human study, you take human donors, you take blood out of them. Even the small things of, are you isolating PBMCs in the same way? Are you storing PBMCs in the same way? Do you want granulocytes? How do you want to store them? So you have all your cells. The first thing you'd want to make sure is that the population that you want to, or the sample that you want to profile, is prepped in the same way. And once you've done that, when it comes to the cytometry aspect, you definitely want to look at antibodies, having good positive controls to make sure that your antibodies are all working, having a good titration where you can clearly delineate between your positive and your negative, using the same lot of antibody throughout the study. Those would be very useful. And when it comes to, before you order your antibody, creating your panel, which is very, very important. So where do you allocate your markers? So if you have something that's very bright and you have abundance of those proteins, you don't necessarily need it on 165. You can put something that's a bit not as bright, like a cytokine, but it's a very important one that you want to put onto your more sensitive channels. So using the resources that are available, the community that's available across the US, and speaking with all your collaborators to understand that your panel, you want to design your panel as best as possible, knowing the proteins in your panel. What is a high abundance protein? What's a low abundance protein? And putting them into the respective channels based on the abundance. Because you don't want, as with flow cytometry, you want something that isn't that abundant to be on a brighter fluorophore. So the same way, you want something that's not as abundant to be on something a bit more sensitive. So you want to look at how you design your panel and then titrate.
And speaking of an antibody panel, one of the techniques people have been using is to create a large batch of their antibody cocktail and either lyophilizing it or freezing it. Do you have any experience with either of these techniques?
So we haven't yet done that. We have heard good results with those who have lyophilized. However, we haven't done it ourselves. We usually, if we have a large study, what we have been doing is running it all, labeling it on the same day.
Sorry, if you have a large study?
We just label it all on the same day. So you make one.
Oh, I see.
Yeah.
And so the samples are frozen, I guess.
The samples are usually frozen.
Okay, yeah.
Yeah.
And you mentioned batch effects. Do you have any experience with batch effect correction or with using reference spike-ins or reference samples?
Yes, we have spoken at length about batch effects.
Yeah, that was not a trick question.
So yes, definitely. Everyone knows, I think you learn a little bit along the way. And some of this is unavoidable because when you have a multi-center trial or multi-center experiments in collaborations, it's hard to get every single thing in complete alignment with each other. So you might have some level of batch effect. So yes, we've had issues with batch effect. And one of the ways in understanding if there is a batch effect is to spike in a reference sample, which is again based off of Cartier's paper and publication. So we do usually do that. It's a very useful technique in understanding if whatever shift you're seeing is an actual shift with regards to the intensity of the marker, or if it is something that's because you have a batch effect. Correcting the batch effects, though, is a much larger challenge.
Yes, and we actually had an interview with we had an interview. The first episode of Single Cellmates was with Sophie, who created Flowsum, and we had a conversation about batch effect correction there. So viewers, you're advised to go and check that interview. So all of what you're describing, to be a bit blunt, is a lot of work.
Yes.
And eventually our goal, if I understand you correctly, is to take this and move it into a clinical trial. Do you think that this level of optimization is scalable? And could it be done in the context of a clinical trial? And if yes, what do you think are the right way to do it?
So I haven't taken part in any clinical trials, so this would be my opinions versus something I have done. But I do think, yes, we can take it into clinical trials because right now vendors are offering lyophilized antibodies, lyophilized panels, which then helps you. The part about is there variation in the master mix and the antibodies, all of those can be taken away. So you can definitely have a situation where you have, as with the flow cytometry, which has been traditionally used for years together, you have the option of, with mass cytometry, a higher level of profiling that can be done.
Yeah, and thankfully there's work. People are thinking about that. There's Peter Brodin, there's Adib Rahman in Sinai, there's Sean Bendel. All of these people published papers recently that discuss workflows for scalable, high complexity studies.
Yes, and we definitely have tools available to be able to scale them as well. So.
Yeah, and I appreciate what you said about connecting with the community and discussing these questions with other people that have dealt with similar experiments.
Yeah.
For my last question, I was wondering whether we can take a glimpse at the future. For you, what is the most exciting thing in the field right now? What are you most looking forward to?
So I have been enjoying a lot of understanding because this brings a wealth of immunology into your day-to-day life because it's a high-dimensional immunology tool. And I've been enjoying understanding how the immune system actually affects how your body responds to anything and everything. So with regards to my specific interest is predominantly in infectious diseases. I have also recently been doing a little bit more autoimmune as well as cancer work, but predominantly infectious diseases. So in the future, what I would be looking at, if you have a global disease or if you have local to your own region, so if you have your own endemic, what would be the differences that you would see in response to the bacteria or the virus in question, and how can we use those differences to our advantage to alter therapeutic or to have the intended therapeutic outcomes in our patients. And ultimately, all of this is targeted towards better patient care.
Yeah, thank you for that. And I agree. It's amazing how the immune system ends up everywhere.
Yes.
I mean, infectious disease is a pretty straightforward application, but we're seeing it in cancer, of course. We're even seeing it in psychiatry and behavioral sciences. So it really is
All the
everywhere.
autoimmune diseases, mediated ones,
Yes.
definitely it's there everywhere. And I think we're just getting into the fields of immunology and how you do find the immune system differences which trigger different responses in different patients.
So Susan, thank you so much for joining us today and for sharing your inspiring immune monitoring work.
Yes, thank you so much for having me, Alad.
Yeah, my pleasure. I would also like to thank our editor, Heather Dwyer, and Astrolabe designer-in-chief, Mihai Koman. Please make sure to subscribe to us on YouTube and LinkedIn to get updates on future episodes. Additionally, check out the Immune Monitoring Biweekly, a regular newsletter with the latest news in the field. Find links in our video description. Thank you and have a wonderful day.